Okay, over the last 10 or so years, it seems like scientists have have discovered a new organ in the human body. This new organ is the microbes in the gastrointestinal tract. We've always known that these microbes are present in the gastrointestinal tract, and they've been with us for thousands and thousands of years, ever since the beginning of mammalian evolution. However, we are just beginning to understand the importance of these microbes in the gastrointestinal tract, largely because now, in the last 10 years, we've developed some new techniques how to actually look at these microbes, not using culture-based technology. So we have this term called the microbiome, and I'd like to define this for you. It's the full collection of microbes, bacteria, fungi, viruses, etc., that naturally exist within the human body. Just last April, the National Institutes of Health in the U.S. Have put out a large request for applications to study the human microbiome and its relationship to disease. And so if you look at the notice that the National Institutes of Health put out, this is what they said. Initiatives in this area would focus on developing a deeper understanding of these communities of microbes in order to determine how they affect human health. Now, what about these microbes in the gastrointestinal tract? When you look at the gastrointestinal tract from the very top, the stomach, we have very few microbes. Most of the microbes are like Helicobacter pylori. You go further down, you start getting an increase in the microbes. And the largest quantity of microbes are found in the distal small intestine and in the colon. And in fact, we have 10 to the 12th microorganisms in our gastrointestinal tract at any one time. 10 to the 12th to 10 to the 13th microorganisms, that is 10 to 100 times as many cells as we have present in our entire body. And the number of genes that these microbes consist of is also 10 to 100 times as many genes as are present in our entire body. It's thought that our body has around 30,000 genes total. The microbes in our gastrointestinal tract has at least 300 genes. So I mentioned before that one of the reasons why there's so much enthusiasm in the last 10 years is because now we know how to look at these microorganisms better. We can only culture approximately 30% of the microorganisms in our GI tract. But using these new techniques, there's about 7 or 8 of these techniques, and this is a picture of what these techniques look like. You take the microbes in the GI tract, and you make DNA from them, and then you put them onto a microarray, and you can actually look at the entire population of genes from the microbes in the gastrointestinal tract at any one time. Now, what we're thinking of is that we usually consider that the microbes in our gastrointestinal tract are pathogens. But they're a lot more than that. They're not just the good and the bad, or they're not just the bad and the ugly. There's also good microbes in our gastrointestinal tract. And we're finding out more and more that if we don't have these microbes in our gastrointestinal tract, it can cause a lot of difficulties. And I want to go through some of this. I also want to, as we go through this lecture, I want you to remember this, that a lot of manipulation of the microbiome, of the microbes in the gastrointestinal tract from early on might actually cause problems in later life. Okay? So think about that as I go through the lecture. Now, let's go back a little bit historically. Back in the early 1900s, these two men, Dr. Paul Ehrlich and Ilya Mechnikov, did some very interesting work. Dr. Paul Ehrlich worked on the immune system. He wanted to find out how the body fights infections, and one of the things that he discovered was the proteins in the bloodstream that are involved with fighting off infections, the immunoglobulins. And Dr. Mechnikov found that white blood cells were important. Now, there was a lot of argument about which of these people were actually right, but they were actually both right, and both of them ended up winning the Nobel Prize. But Dr. Mechnikov went to do some other very interesting work. And one of the things that Dr. Mechnikov did was he traveled to this country in Eastern Europe, Bulgaria, and he found that people in Bulgaria, certain parts of Bulgaria, were living to very old age, 80, 90, 100 years of age. And he wrote a book called The Prolongation of Life. And with this book, in his book, he stated that one of the things that he found that these people were eating a lot of was fermented milk products. Well, he looked under the microscope at some of these fermented milk products and found that there were microbes in these fermented milk products. And in fact, to this day, there's actually a microbe named after what he first discovered, Lactobacillus bulgaricus, coming from the country Bulgaria. Well, now there's a lot of information about yogurt and fermented milk products, and it's thought that these foods play a tremendous role in terms of our health. What I want to do is talk about some of the potential benefits, but also want to talk a little bit about some of the downsides of using these in babies, using microbes in babies. First of all, I want to talk about the microecology of the gastrointestinal tract, its relationship of the intestinal microbes to the innate immune system and the inflammatory pathways in the intestinal tract, and relationship of the intestinal microbes to protection of the GI tract, and then the role of probiotics, live versus dead probiotics, prebiotics, and also a new term, postbiotics. Now, some of the beneficial effects of commensal microbes, these are the microbes that live in our gastrointestinal tract, have to do with nutrient metabolism. I'm sure you're all aware of the fact that vitamin K is produced by the microbes in the gastrointestinal tract. If you do not have microbes in the gastrointestinal tract, you do not have very good vitamin K production. Okay? Tissue development, resistance to colonization with pathogens, and maintenance of intestinal homeostasis. Now, let me just give you some information that shows some of this importance. This is work that was done at Washington University in St. Louis by Dr. Gordon's group. And what they did was they looked at the intestinal villus development, and here we see intestinal villus development on day 14 postnatal in these mice and day 28. And what they did was they stained the intestinal villi with this green substance that stains the capillaries. And here you see this is just normal growth of capillaries. A lot of growth here by 28 days compared to 14 days. Now, what happens if you take these animals and grow them in a germ-free environment where there are no microbes in the gastrointestinal tract? Okay? So here's what they did. For 40 days after birth, they grew these mice in a germ-free environment, and here you see the villus capillaries after 40 days. Here's the situation where they grew them in a germ-free environment for 30 days, and then for 10 days added conventional microbes. They took them out of the germ-free environment, put them into an environment that had microbes, and you see that major increase in the development of the capillaries. And The same thing over here with only 10 days of microbe exposure, but this is only with one microbe. This is called Bacteroides theta iota omicron. Only with one microbe, you get massive development of these capillaries compared to here. So, microbes in the gastrointestinal tract, at least as far as capillary development, seems to be very important.